previous lecture we started a new topic, electrophoresis, and we discussed about the general principle of electrophoresis. We can little bit revise that. So, when a charged particle moves under the influence of an electric field, that particular phenomenon is called electrophoresis, and a charged particle will experience a particular force and it will move as per its charge towards the opposite electrode. And so, there is one which is the force because of the electric field, and another will be the frictional force because of the particle moving with a speed through a media, and that particular medium will pose a frictional force. Together both of these will will lead to uh, the resultant electrophoretic mobility. So, as you have seen, the three main factors which affect a electrophoretic run, that is the one that is electrophoretic mobility of a particular molecule, then the heating problem which is also has to be solved to a certain extent, and then third was electroosmotic flow which might happen due to the support media. And as we have seen, the electroosmotic flow, the problem of that has been solved to a great extent in gel electrophoresis. Now, in this lecture we are going to extend our discussion on electrophoresis, and we are going to discuss about the gel electrophoresis. Now, what are the main components of gel electrophoresis? Before we go into that, as we will go along, we will be going to explain in this lecture that there are two kinds of mainly gel electrophoresis techniques which are widely used. One is horizontal gel electrophoresis and another is vertical gel electrophoresis. Horizontal gel electrophoresis, the media used is agarose, and in vertical gel electrophoresis the media or support matrix is used is the polyacrylamide. All right. So, let us start with the main components of gel electrophoresis. Now, the main components, one, like we were discussing, the gels or the support media, or we can call it matrix, and as I said, the most widely used matrices are the agarose and polyacrylamide. Then with this support material, another thing you require is electrophoretic apparatus or chamber with buffer reservoir. So, we will show you that in a little while. Then electrodes in the buffer reservoir, that is positive electrode and negative electrode. Then you require a power supply for particular power setting, actually, that is you can run it on constant power, or you can run it on constant voltage, or you can run it on even constant current. The power supply could be like it could give you a power of 200 to 500 volts, or say 400 microampere to 100 milliampere, and so. So, these are essential components of a particular gel electrophoresis apparatus, that is electrophoretic apparatus, power supply and the gels. Now, there could be three kinds of gels like mini gel, midi gel or maxi gel. Mini gels are very small, which could be like say 8 centimeter by 8 centimeter, and the larger gels or the maxi gels could be anywhere in 40 to 20 centimeter. So, and midi will be in between them. So, there could be three kinds, and depending on your application you can run either. Most of the time mini gel is the most routinely used, other gels could also be utilized as per the requirement. All right. So, let us start with support media. Now, the pioneering work on electrophoresis, as we have discussed by Tselius and co-workers, was performed mostly in free solutions, but many of the problems associated with this approach like the adverse effect of diffusion and convection currents, and it could be these things could be minimized by stabilizing the medium, and this was achieved by carrying out electrophoresis on a porous mechanical support. which was then wetted in a electrophoresis buffer, and in which electrophoresis buffer ions and sample ions electrophoresis could occur. So, like this problem of diffusion and convection currents could be minimized in a support medium, and so therefore, it was needed that from free solution one moved to a support media. Now, earliest support used were like filter paper or cellulose acetate paper strips were utilized. They were wetted in electrophoresis buffer and they were the electrophoresis was run. But nowadays these media are very infrequently used, and this has been done because of the introduction of and use of gels materials, which led to a rapid improvement in the methods for analyzing macromolecules. Now, gels are the most widely used, and like I said, agarose and acrylamide gels are the most widely used matrices. Now, if you consider a gel structure, so gels structurally are intermediate between solids and liquids, and the structure of gels as well as their interaction with macromolecules is not so well understood. And this is like mostly the electrophoresis, which like when molecules run through the porous structure where sieving effect takes place. It is a quite unknown area actually. Now, it is difficult to cast gels of precisely controlled structure, because like I said, it is a random structure with pores. Now, two most important and widely used matrices, like I was saying, are agarose and polyacrylamide. Now, in the process of gelation of these two gels, the individual polymers of both gels combine into fibers and then aggregate into larger bundles joined in random meshworks. So, both gels they exist as random distribution of solid material and pores, which we can call open spaces here, and they are apportioned in say Poisson distribution of sizes. It is like you have a mean of sizes, but they are not like exact equal pore sizes which you obtain when you cast gel every time. So, this has to be understood that these are kind of meshwork which is which combines and aggregates together in a very random fashion, and their pore size you can say is a mean of a certain value, but they are not exact pore size. So, travelling of macromolecule through them is also like you have to, if you want to say compare the two gels, then you have to do it through running standards, and you might not be able to reproduce the results exactly because of this problem, because gel every time you will cast a gel it will be different. So, the idea of gel pores is quite imprecise, as I was saying, and only refers to the resistance which a gel imparts to the migration of charges, which varies in a shape and size also. So, you have one is the charge, another is shape and size will also play a role because of the pore size of the gel. Now, so gels can be considered as three dimensional sieves actually, and what happens is that molecules during electrophoresis move through these buffer filled pores of the gel, and the ability of macromolecules, say proteins or DNA in particular, to migrate through these different size pores or pore regions will depend on the structure of the molecule. So, if you have a larger structure and particular kind of shape, maybe the resistance is more to the movement as compared to the smaller molecule and a particular shape, that resistance might be less. So, this will certainly affect the migration patterns and the velocity at which these molecules will move. So, the gels are a particular, you can say, three dimensional sieves, but the movement migration will certainly differ from gel to gel. Now, there are advantages of gels over free solutions, as we have discussed earlier. The porous gels, or they will act as sieves, uh, and so they limit diffusion of the sample molecule. Like I said, electro-osmotic flow is minimized or suppressed. Likewise, Joule heating is also quite suppressed in gels, but still the heating do generate, and many times cooling apparatus, cooling systems has to be put in to reduce the heat. Uh, the separated molecules will segregate into distinct or discrete zones corresponding to their electrophoretic mobilities when you are running them in these gels, and at the end of the electrophoresis, when electric field is turned off, individual analytes are constrained at their final position as a distinct band pattern, which can be visualized by say staining or certain other positions. Now, this one, like here, when you have a free solutions, certainly diffusion will occur, and if you leave it for a little longer, then bands might not remain as sharp bands. So, gels have their properties, and gels have a particular way to like to separate and analyze the samples, and they solve many problems which are faced when you work in free solutions. Now, like we were discussing, there are two widely used gels. One is agarose gel and another is polyacrylamide gels. Now, let us discuss first agarose gel. Now, the pores of the agarose gels are very large, or you can say larger as compared to polyacrylamide gel. So, agarose gel electrophoresis is the easiest and a very common way of separating and analyzing DNA. DNA are large molecular weight molecules, and as far as proteins are concerned, proteins, if they are very large, like say above 500 kD or so in size, then they can be separated in agarose gels. Otherwise, polyacrylamide gels will be utilized. But for proteins, agarose gels are also widely used for certain applications, like for example, immunoelectrophoresis, where they only act as anti-convecting support media, and where protein movement is not hindered, and antibody antigen interactions are seen. So, you are not really separating the protein, but interactions you want to see. So, immunoelectrophoresis, the agarose gels could be utilized. Polyacrylamide gels are used for the separation of the proteins, which are a smaller protein molecule as compared to what is used in agarose gel, and also very small DNA fragments could be run on polyacrylamide gels. Like I said, the pores on the polyacrylamide gels be smaller. So, large molecular weight DNA fragments cannot be run, but small DNA fragments could be seen, like say in DNA sequencing gels are polyacrylamide gels, and where you have small fragments of DNA, where you are trying to sequence in earlier times when gels were utilized for sequencing the DNA, the polyacrylamide gels were utilized. The gels can be used for the simple visualization, they could be utilized for quantification, or even to isolate a particular band of DNA molecules. Now, most of the time the gel electrophoresis is an analytical technique, but it could be made preparative, and small amounts of samples, particularly the nucleic acids, could be purified from there, and even proteins could be separated or could be extracted from the gels for certain applications, like protein sequencing or N-terminal sequencing, or say internal sequencing by mass spectrometry. Now, in agarose gel, the DNA could be visualized in the gel by addition of ethidium bromide. So, this binds strongly to DNA by intercalating between the bases and is fluorescent, meaning that it absorbs invisible UV light and transmits the energy as visible orange light. So, when it intercalates, it fluoresces, and which could be seen and could be visualized here. So, Agrogels are mostly used with the DNA or nucleic acid. Now, agarose is a linear polysaccharide obtained from the red algae or certain weeds, and it is made up of the basic repeat unit, that is agarobioes. And agarobioes comprises of alternating units of galactose and 3,6 anhydrogalactose. So, it could be like several algae is the source of this, and it could be purified from there, extracted and purified from there. Now, this agarose is supplied as a dry powder, and agarose varieties differ in their physical and chemical properties, like gel strength, gelling temperature, porosity, electroosmosis, which depends on the purity of the gels, like if it contains certain charged groups on its surface, then certainly electroendoosmosis problem will occur. Now, most agarose gels are made between say 0.7% and 2%, or it could go up to 3% also in certain cases. 0.7% gel will show be utilized for good separation of large DNA fragments, like say 5 to 10 kb fragments, whereas 2% gel will be good for smaller fragments, which are much smaller, like say 0.2 to 1 kb fragments. So, depending on what kind of fragments you are going to separate, you can cast the gel. Mostly 1% gel is very common in the agarose gels. Now, this is simple schematic of the two units here, which agarose is made of, that is the repeating unit, agarobioes. Now, the gelling properties of the agarose are attributed to both intra and intermolecular hydrogen bonding within and between the long agarose chains. So, this is like the whole matrix which is formed is due to the hydrogen bonding pattern actually. How this gel is cast actually? It's a in a very simple way. How do we make the gel, agarose gel? Now, this is a very simple, like what is done is you weigh out a required amount of agarose. Like I said, it is supplied as a dry powder. So, if you want to make 1% of gel, then you weigh 1 gram of gel for 100 ml. You will weigh the required amount of agarose in say conical flask, and you will add required amount of buffer, which is TBE or TAE buffer, and you will mix it, swirl to mix it. Now, what is done is you will microwave this for say 1 to 2 minutes, and the agarose will be dissolved, it will be mixed with the buffer. You will then leave it to cool for a very small period of time, a few minutes, and temperature will be little down, like say around 60 degree or so. So, you can add then ethidium bromide to the gel. Now, ethidium bromide here could either be added to the gel, say 1 microliter of ethidium bromide, which is 10 milligram per ml is enough, and you can swirl it, or it could be done that you can run the gel without ethidium bromide also, and you can stain the gel afterwards in a solution containing ethidium bromide. So, both ways it could be done. So, once you have added the ethidium bromide, if you want to, then you will pour the gel slowly into the tank, that is the casting apparatus. I will show you that how it is done. You will push any bubbles or anything and through, like say you can use tape or certain thing. Then you have to insert the comb, and the combs are inserted so that they can make slots for sample to be loaded. Like when gel solidifies, this melted gel when solidifies, then if the comb is there will be slots or wells created where you can load your sample in the gel. You have to leave it for say at 30 minutes or so, or say 1 hour for solidifying, and then finally you will it is ready to be used. You will pour the, you will put it in the gel running apparatus with electrodes, and you will fill the gel tank with the TAE or TBE buffer, whichever you are, tris EDTA or tris borate buffer. So, you will tris acetate EDTA or whichever buffer you are utilizing, and you submerge the gel in that buffer, that is running buffer. Here it shows very clearly how this is done. So, before this figure, I would like to show you how this whole process has been done here during the casting of the gel. All right. So, what I was talking about is, first thing is, you take a, say, a conical flask, and this conical flask you will weigh a particular amount of material, that is your gel, and you will fill it with the buffer. All right. Now, this gel is still solid gel, it will settle down here, you will swirl it and you will put it into the microwave. So, means you have to heat at this conical flask, which contains the weighed amount plus buffer, weighed amount of agarose and the appropriate amount of buffer. Once you put it in microwave, the agarose will melt and it will mix properly, like you will see the melted buffer, a thick melted solution. Now, after that you will cool it for a while, and then you can add ethidium bromide. Now, you will have a gel apparatus. Now, this gel apparatus will have say from two sides it is enclosed, it is it is a glass strip with two enclosed sides, and other sides you can tape it actually. So, when you put the tape, you can tape this from both sides, then what will happen? It will be enclosed from all sides. Now, this material you can pour this material in here through, you can put a glass rod, or you can there could be a glass, through glass rod you can pour, or very carefully sides you can pour this. So, what will happen? This will evenly distribute onto this gelling apparatus. Now, once gelling apparatus you have put in, if there is any bubble you can see, you can push it or you can remove it through tip or otherwise. Once it is done, there is a comb. What is comb? Comb something looks like this. It is, these are slots here. It looks something like this. So, these combs, like this will not, it will like go into the gel, and this area which is like, you can say this, these areas will be vacant here, or wells will be created. So, this comb can be placed in here in horizontal in one position, and this comb here, when it is placed in here, then it will create wells. Number of wells will be according to the number of these how many number in that particular comb it contains, like strips it contains actually. Now, once you have done with this, then your gel is ready after say 1 hour, and it is ready to be used. All right. So, let us go back to our discussion. So, here if you can see in this figure, like I was showing, gel is casted here, and you can see here these are the wells which are a few numbers here. Now, you have a DNA sample, and there could be more than one DNA sample, or there could be like different samples here, or one sample loaded into each well. Now, this sample could be treated, and you can get like say this has been this gel sample has been restriction digested, and you what is created here from one DNA sample you have got many DNA fragments. Now, these DNA fragments needs to be separated here. So, what will happen? You will, with a pipette, you will load this into here. Now, when we are loading this, the loading is done in a sample is prepared in a say certain high density solution, like you can have sugar, sucrose solution, or maybe glycerol or something else could be utilized, so that the your sample settles down and does not float or diffuse in the buffer. So, it settles down at the bottom of the well. Then there are two electrodes here, negative and positive, it is run in the gel apparatus, and what you get is a bending pattern, and this bending pattern where what will happen that your DNA fragments, since they are negatively charged, they will move towards the positively charged electrodes. The smaller DNA fragments will move faster as compared to the larger DNA fragments, and on that basis separation will take place. This you can analyze in UV illuminator, and or you can even cut the gel and you can get the DNA extracted. So, agarose gels are used for electrophoresis of mostly nucleic acids, but you can also use them for proteins also. Like I said, for proteins the pore size of 1% agarose gels are larger relative to the sizes of proteins, and agarose gels are therefore used in techniques like immunoelectrophoresis or say isoelectric focusing. Likewise, they could be used where proteins are moving unhindered. There are other aspect of, like when you are trying to purify the or extract the DNA material from the gel, agarose gel, then low melting temperature agarose gels are available, which is like they melt at say 62 to 65 degrees Celsius, and they could be very useful to extract the separated DNA samples, and you can recover and purify those in purified form very easily, like without you have to in other gels you might have to melt them at a higher temperature, and recovery is low, but in here the recovery is very high. Now, because of poor elasticity of agarose gels, mostly the electrophoresis done in horizontal gels. So, because of this problem in agarose gels, it is horizontal gels and not vertical gels. All right, this was about the agarose gels. Now, the second most widely used gels, and particularly for proteins gels, these are polyacrylamide gels, and they are routinely used in many teaching labs and research labs. So, the electrophoresis performed in acrylamide gels are frequently referred to as polyacrylamide gel electrophoresis or PAGE, PAGE. Now, here like if you could recall in agarose, polymerization was done like you melt and then you solidify, and through hydrogen bonding the whole structure or like matrix structure is formed. Here copolymerization of acrylamide and N prime methylene bisacrylamide is performed. So, bisacrylamide is two acrylamide molecules linked by methylene group, and they are used as a cross linking agent. So, what is done is that acrylamide monomer is polymerized, and they polymerize in a head to tail fashion, and so the long chains are so the long chains are formed, and in these long chains insertion of bisacrylamide occasionally into the growing chain results into a cross linked matrix of fairly defined structure. So, the polymerization of acrylamide is an example of free radical catalysis. All right. So, this figure here shows you this is acrylamide molecule, and what happens is that this is bisacrylamide and long chains of acrylamide are formed, and like these two chains, and in between bisacrylamide gets inserted. As this cross linking occurs, depending on the concentrations, the pores will be created. So, polymerization of acrylamide is an example of free radical catalysis, and in here polymerization is initiated by the addition of ammonium persulfate, which acts as an initiator, and timid as an accelerator. Timid is tetramethyl ethyl ethylene diamine. Now, timid catalyzes the decomposition of the persulfate ion to give a free radical, a molecule with unpaired electron. Now, these free radicals are highly reactive species, and they will catalyze the cross linking in head to tail manner. So, what happens is, if we can see here on the screen, so you have a persulfate ion, and through so what it is decomposed into, here there is a single unpaired electron. So, free radical is generated. Now, if M is the acrylamide, it represents the macromolecule, and R is the free radical, then there is a generation of, it is a kind of chain reaction which leads to the formation cross linking in head to tail manner. So, it goes on actually, and in between bis acrylamide is introduced. Now, here one has to remember that, like it is a free radical reaction, gel solutions should be degassed for proper polymerization, like oxygen will remove free radical and it might hamper with the polymerization. Apart from this persulfate and timid polymerization, there could be photopolymerization could also be utilized. It can be alternative method that can be used for polymerization of acrylamide gels. So, ammonium persulfate and timid are replaced by riboflavin. Now, when gel is poured, it is placed in front of bright light for say 2 to 3 hours, and photo decomposition of riboflavin generates a free radical that initiates polymerization. So, both ways you can do polymerization of acrylamide gels. Now, polyacrylamide gels are characterized in terms of percent T, which we call weight percentage of total monomer including cross linker. So, here acrylamide monomer is T and cross linker is C. So, percent C is the proportion of cross linker as a percentage of total monomer. So, percent T means it carries both monomer as well as cross linker. Percent C is the proportion of cross linker in the total monomer. So, the pore size in the gel can be varied by the changing the concentration of both acrylamide and bis acrylamide. So, what happens is, when percent T increases at a fixed low concentration of percent C or cross linker, then the number of chains increases and pore size decreases. Now, when percent C is varied at constant percent T, then pore size decreases to a minimum at about 5% C, but it then increases with further increase in percent C. What happens is, it can be due to the formation of shorter and thicker bundles of linear chains of polymer. So, you can increase the cross linker to a certain extent only. Now, monomer solutions are made as concentrated stock solutions, and so from stock solutions you will make various percentages of acrylamide gels. Like for protein separation, 30% T and 2.7% C stock is preferred. For nucleic acid, 30% T and 3.3% C cross linker are used, and the final concentrations of say 0.05% of ammonium persulfate and timid are used for resolving gel. We will be discussing what is resolving gel, and for stacking gels 0.05% APS and 0.1% timid can be used. So, these are different various concentrations, and every lab standardizes its through protocols the running conditions for electrophoresis gel. Now, acrylamide gels can be prepared between 3 to 30% acrylamide. The low percentage gels with larger pore size are used for free migration of proteins, like for example, simple IEF or stacking gel in SDS page. Both techniques we are going to discuss in later lectures, or to separate DNA molecules. Gels between 7.5 or so and up to 20% are used for the separation of proteins, whereas smaller pore size introduce a sieving effect and proteins are separated on the basis of charge. So, most of the acrylamide gels are vertical gels, and vertical slab gels are very common, where many samples can be run in identical conditions, and they are the routinely used apparatus for the analysis of proteins as well as for separation of smaller DNA fragments, say in DNA sequencing gels. Now, there are tube gels also which are used quite a bit, but only problem is in tube gels only a single sample can be run at a time, and can be this particular tube gels are used for as a first dimension for 2D gels. So, that could also be one. I will be discussing 2D gels also in a while. All right. So, this was little bit about that what are the different aspects of polyacrylamide gels. Now, how do proteins migrate through the gel, or how this acrylamide gels are run actually. So, when you have casted the acrylamide gels, let me show you here on your screen how this whole thing works here. So, as we have seen for agarose gels, the acrylamide gels a little bit require more processing here, and like I said, the first thing is that you have a stock solution actually. So, you have a particular stock solution. Now, this stock solution, like I said, it is 30% acrylamide with say 3% or so bisacrylamide, and what you have done is you have made this stock solution, you can store it, and you have to make different percentages of gel as per requirement. Like for example, you want to make 10% gel. So, what you will do is, and there are stock solution is of acrylamide, then there will be buffers. You have to take these buffers which are buffers which could be running buffer like electrode buffer, and could be gel buffers where you have to make the gel, and these buffers we will be discussing later on. There could be like a condition like stacking gel and resolving gel, or it will be a continuous system where a single gel is utilized. So, what you will do as per your calculations, you will mix this, say you are making 10% gel, 10% acrylamide gel, then the final concentration, like, so what you will do is you will take the buffer, you will take certain amount of from stock solution, acrylamide solution, and you will be mixing here so as to that final concentration is 10%. All right. Now, once this is 10%, you will add here the two things, one is APS and timid. All right. Like I said, timid is the accelerator. So, as timid is added, this whole process starts very fast, and we it is a very small amounts are added. Now, once you have added these things to the 10% gel which you have made, and you have already added buffers and all, then very quickly you have to pour this into the casting apparatus. These casting apparatus, if I say this is like, these are two, if it is a slab gel electrophoresis, then there will be two plates, one something like this, another will look something like this. All right. So, this plate will be put on top of this plate with spacers here. Spacers are very thin strips which are rubber or some other. These spacers which are only a few, like say 0.5 mm or 1 mm, they will be placed in here, and this plate will be placed on top of this. So, what you will find, this plate is being put on top, and they are being separated by the spacer. All right. So, there is a gap between the two plates. So, this has to be understood that the two plates, when they are being put on each other, they have the gap because of spacer. Now, what will happen? This material has to be poured in here. So, you will pour your material in here, and you have to be very careful through the walls you will be pouring this. Now, if it is a single gel, like only resolving gel to the top, you will put it to the top. If it is a stacking gel and resolving gel will be put first, say up to this height, and then stacking gel will be put in here. All right. So, this we will be discussing later on. Once you have done this, then remove any air bubble. You can put say a layer of organic solvent on top of it, just to make a uniform shape here, rather than disturb bed above bed, and then finally it will solidify very fast in say 15, 20 or 30 minutes. Once it is casted, then it will be put into the running apparatus, which is contains electrode, place for electrode buffers and other putting that. Now, here before like as you have poured this, you have to put combs. Again the combs, as I have shown you in agarose gels, this comb will be put in here, and this comb will create the wells in here in the gel, so that you can load your samples. All right. So, let us go back to our discussion. So, how do you run the proteins on this and how do they migrate? So, what is done is say a viscous protein solution is layered on top of the gel in a small well, as I told you how well is created, which are molded into the gel during the polymerization process. Then bottom and top parts of the gel are inserted into reservoirs containing a buffered solution and the appropriate electrode. The electric field is applied and the proteins migrate through the hydrated gel, and the components of the buffer must not bind to the protein for a good run. The pH of the medium must be such that proteins have the appropriate charge. Like so, you want to transfer them from say negative electrode to positive electrode. So, they have to have a particular pH, particular charge, and that will be done as per the pH of that particular buffer. Equipment for this, like I was showing you, to conduct paste is very simple. It consists of a mold to form the gels, that is a casting one, then an apparatus to hold the gel and buffer, and power supply, as we were talking about, to capable of delivering the required voltage or current. So, typically this is how it looks like actually. If you see here this particular one on the screen, this is tank buffer, that is the top tank buffer and the bottom tank buffer. So, these here contains the electrode buffer, or you can call it running buffer. As this tip is showing here, through pipette you can load your samples in these wells very smoothly, so that it does not diffuse out and it settles down into the well. And this is your gel which has been casted, wells have been created by your comb, and as you switch on the electric current, they will start. So, if it is a negative electrode and this is positive, all negatively charged species or protein samples will move towards the positively charged electrode, and according to their size and the charge they will separate out. So, remember the smaller protein molecules will run faster and the larger protein molecules will run slower because of the resistance of the gel depending on the pore size here. So, that is how the whole separation will occur, and we are going to discuss this quite a lot as we go as we discuss STS page and other techniques in here. So, this is a very simple electrophoresis or vertical gel electrophoresis apparatus. Likewise, tube gels could be run in here, where rather than a slab gel or this plate gel, you will have a tube and only single sample could be run. The whole apparatus looks something like this. You have enclosed buffer chamber and these are the electrodes, 2 electrodes here. The gel apparatus is kept in here, and as you switch on the electric current, this whole pattern will the protein migration will take place and separation will take place. After the protein has been run, you can remove the gel plates from here. Gel plates could be separated and the gel could be put in the staining solution, like say solution containing commercial or any other dye, and they could be visualized. So, all right. So, today we have discussed about vertical gels and the horizontal gels. The horizontal gels are agarose gels and vertical gels are polyacrylamide gels. We have learned today about how they are casted and how they are polymerized and how they are run, what different kinds of samples are being run on these gels and details about that. In the next lecture, we are going to start with say STS page technique, and also since modern STS page is a discontinuous system, we will little bit deal about continuous and discontinuous buffer systems. So, we will end this lecture here. Thank you.